So Paul, who will be the lead speaker for this episode 3 of the webinar series, is a principal scientist within the Biologic Solutions group here at Waters. Paul's focus is on biomolecule separations using ion exchange and size exclusion chromatography. Before we get into the episode 3 and Paul's portion of the webinar, I'd like to go over a few housekeeping items. You'll notice that you have a toolbar at the bottom where you can put things like your questions and answers, your questions that you have for us, and we'll answer them at the end of the presentation. You'll notice that your microphone and cameras are switched off by default. And like I said, if you have any questions for us, please contact us at any time by with the question and answer button. And I'd also like to remind you, for the purposes of resharing and reviewing the webinar series afterwards, we are recording the webinar today. With that, I'd like to turn it over to Paul Bigos.
Thank you, Jim, for the introduction, and thank you all for joining the 3rd episode of our biosimilar webinar series. My name is Paul Bigos, and I'm a scientist in the QA/QC Biologic Solutions group at Waters. And in this presentation, we'll be talking about deploying new technology to reduce the burden associated with migrating methods downstream in the analysis of mAbs. On the agenda for today's presentation, I will introduce Waters' MaxPeak High Performance Surfaces technology and mention where we've incorporated this technology for improved analysis of biotherapeutics. I will then give an overview of method scaling and show examples of how it is applied to charge variant, size variant, and peptide mapping analysis. And lastly, we will talk about method migration and take a look at migrating a method from a legacy instrument to a modern instrument for peptide mapping. To calibrate the audience on some of the systems and columns we'll be using in this presentation, MaxPeak High Performance Surfaces are a new and innovative technology that's designed to increase analyte recovery, sensitivity, and reproducibility by minimizing analyte to surface interactions. So this is a hybrid organic-inorganic surface that forms a barrier between analytes containing electron-rich functional groups and metal surfaces along the flow path. So functional groups such as phosphate, amine, and carboxylate groups are known to absorb onto stainless steel components, which consequently cause recovery, resolution issues, and method variability, which can pose data quality and regulatory risks. To address these risks, we've incorporated MaxPeak HPS surfaces in several products to improve detection limits and reproducibility, starting from sample preparation all the way to detection. So this technology was first introduced in our Quan recovery vials and plates, which reduce analyte losses from nonspecific binding, and most recently have been incorporated into 2 of our systems, the Acuity Premier UPLC system and the Arc Premier UHPLC system. As a total solution, MaxPeak HPS surfaces have also been incorporated into a comprehensive family of columns to offer a range of selectivity and particle size choices, as is shown in the slide. So in tandem, all of these solutions are holistically designed to work together and eliminate analyte losses caused by metal interactions and act as a universal platform for separation of biotherapeutics. By utilizing some of these products, you can expect to see increased sensitivity, reduced variability, and reduced passivation and conditioning time prior to your analysis. In case you missed last week's episode of the Biosimilar webinar series, our colleague Samantha Ippoliti presented on a characterization at the intact, subunit, and peptide level of biosimilar mAbs using the Xevo G3 QTOF mass spec. In this week's episode, we will be talking about migrating methods from these characterization labs where all the quality attributes are identified, and then migrating them to manufacturing environments where they will take on more of a routine analysis role. These workflows tend to transfer from MS-based methods to simpler platforms for routine analysis. For the workflows we'll be going over today, we will be transferring methods from the Acuity Premier system to the Arc Premier system. And for some of the listed upstream workflows, LCs are engineered to provide the highest sensitivity through smaller column particle sizes, which are usually less than 2 microns, and smaller tube and column inner diameters, and capabilities to operate at higher pressures. And as analytical processes are refined and the potential quality attributes are identified in some of these listed upstream workflows, methods will eventually have to be migrated to manufacturing environments. So these methods, once they are validated, are typically run on larger bore columns with larger particle sizes to operate under more demanding conditions as a monitoring assay. So to facilitate this transfer, these methods will have to be scaled between systems and columns. But before we go into method scaling, I wanted to take a moment to mention an update to chapter 621, titled Chromatography of the USP. So prior to December of last year, there were no allowable changes to column dimensions for gradient methods for any USP monographs. So with this December 2022 update, several variables can be changed as is shown in the table, with the most significant change being able to alter the column length and particle size. Utilizing these allowable changes, you can now take advantage of modern HPLC technologies and see benefits such as decreased solvage usage, improved method performance, and shortened analysis time without needing a full revalidation, only a verification of the adjusted method. So now going into method scaling, to properly scale a method, several parameters must be considered to ensure the same separation can be achieved in a downstream system. As you can see in this slide, to successfully scale a method across systems and columns, differences in column geometry, method conditions, and system volume differences must be accounted for. So while these calculations are well understood, they do take some time to calculate and are subject to error as a manual process. To address these concerns, we've automated the scaling process through the Columns Calculator version 2.0, which is available to download on our Waters website. And it also comes as part of the Empower or MassLynx CDS software. So the Columns Calculator is easy to use, and users only need to enter the column dimensions used on both systems and the method conditions of the original method on the left side of the calculator. The calculator will then scale the method based on the calculations shown in the previous slide and automatically populate the recommended method conditions on the right side of the calculator. The only variable that must be experimentally determined is the dwell volume of each system. So if you want to match retention times of your analyte across both systems, then the dwell volume will have to be measured to ensure that the gradient delivered across the column is accurately reproduced at the time of injection. So the dwell volume represents the volume of the flow path from the pump to the head of the column, as is illustrated in the schematic for the Arc Premier system. So this volume changes based on the design of the LC system, and to experimentally determine the dwell volume, a simple gradient from mobile phase A to B can be used with a UV active compound. So in our case, we used caffeine as the UV active compound and spiked it into mobile phase B at a concentration of 10 milligrams per milliliter. And as shown in the chromatogram, the blue trace represents the detector response, and the black trace represents the programmed gradient. So after normalizing the response, we measured the retention time at 50% of the absorbance maximum. And so the difference between the detector response and the programmed gradient was then multiplied by the flow rate to determine the dwell volume of the system. And in general, HPLC systems will have a larger dwell volume than UPLC systems due to the larger ID tubing, and quaternary pumps will have a larger dwell volume compared to binary pumps. So for the data shown here, we found that the dwell volume of the Arc Premier quaternary system to be about 910 microliters. So after experimentally determining the dwell volume on both of these systems, we're now going back to the columns calculator and inputting the information into the system section, and a recommended isocratic hold was automatically calculated. For our case studies today, we experimentally measured the dwell volume on both the Acuity Premier and Arc Premier systems. So on the right side of the calculator, the method has been automatically scaled, and the recommended isocratic hold volume is displayed in the bottom right corner as shown in the red box. The isocratic hold is then incorporated into the instrument method under the miscellaneous tab for the solvent manager. So the default setting is for the gradient to start at injection, but in our example today, we are migrating a method and using an isocratic hold of 785 microliters, which was calculated from the calculator after injection. If we were doing the reverse and we were scaling a method from, say, an Arc Premier system to an Acuity Premier system, the columns calculator would have calculated a negative isocratic hold, which would mean the gradient would start before injection. So moving on to our case studies, we will be looking at 3 different separation techniques that are commonly deployed for monoclonal antibodies. So first, we will be looking at ion exchange chromatography, which is often a salt-based, salt-driven mechanism relying on the charge interaction between an analyte and the stationary phase. Elution is achieved by increasing the ionic strength to inhibit binding of the analyte to the active site of the stationary phase, as is shown in the slide. So an alternative to using a salt gradient is to use pH to elute analytes. So while ionic strength is held constant, the net charge of the protein is manipulated by altering the pH of the mobile phase. So in this presentation, we'll be evaluating both salt and pH gradients for monitoring of charge variance. And next, we'll be looking at size exclusion chromatography, which for MABs is an isocratic technique that separates molecules based on their hydrodynamic radii. Separation occurs when molecules of different sizes are either included or excluded from the porous packing material. And so smaller molecules will pass through these pores and have a longer distance to travel, which consequently means that the molecules are eluted in order of decreasing hydrodynamic radius. And so this technique is commonly used to monitor purity levels by separating the monomer from any aggregates and fragments. And then lastly, we'll be scaling a reverse phase method, which separates a mixture of components based on their hydrophobicity as the concentration of organic modifier is increased in the mobile phase. So for MABs, peptide mapping is the technique of choice to verify primary structure. So for our first case study, we'll be looking at the charge variant profile for an infliximab innovator and biosimilar. We utilize a cation exchange column for separation of charge variants, which means that the acidic species are the variants that elute prior to the main peak, and basic species will elute afterwards. So these variants are monitored throughout the development and manufacturing process and are caused by a variety of factors, to name a few, such as oxidation, deamidation, and glycosylation. So MS is heavily utilized to characterize the quality attributes that could impact product quality, and it's used to identify an acceptable charge variant profile. So then these methods are then transferred from characterization to manufacturing labs, which will be used for routine monitoring by optical detection. So in this slide, we'll be scaling a method from the Acuity Premier system using a 2.1 by 100 millimeter 3 micron column to an Arc Premier system with a 4.6 by 100 millimeter 3 micron column. So looking at the table retention time data, both systems and column formats are able to reproducibly deliver the same profile with retention time RSDs below 1% for the main lysine variants. On this slide, we can also see the impact of the isocratic hold when dwell volume is and isn't accounted for. So when it's not utilized, the peak profiles elute earlier by about 2 minutes, as seen in the middle trace, when compared to the original method. However, when we do utilize the 785 microliter isocratic hold that we calculated earlier, as is shown in the bottom trace, the peak profiles align closely with the original Acuity Premier system method. So when we account for all variables, we can preserve the chromatographic performance in terms of both selectivity and peak area without the need for further optimization. So this is shown in this slide where we compared the relative retention time and peak area percent between the original and scaled method for 7 peaks throughout the charge variant profile. So using peak 3 as the reference peak, the relative retention time was highly similar between systems and columns, with absolute differences below 0.03 for the scaled method. And similarly, the relative peak area percent was reproducible and stayed within 0.5% of the original method. As we discussed earlier, ion exchange chromatography can be a salt or pH-driven separation. So we evaluated the IEX method using a MES salt gradient and a pH gradient using water's bioresolve pH concentrates. As shown in the table, the relative retention time and peak area percent were conserved equally as well across systems when using either the pH or the salt gradient. Moving on to our next case study with size exclusion chromatography, I wanted to briefly introduce our Max Peak Premier line of SEC columns. These columns feature a novel BEH-PEO particle that is coupled with Max Peak HPS surfaces to significantly decrease secondary interactions for SEC. So for the data set on this slide, we used a base 100 millimolar sodium phosphate buffer and incrementally increased the salt concentration from 0 to 200 millimolar. From the chromatograms, we can see that the Premier column has significantly improved recovery of the monomer and the high molecular weight species at lower salt concentrations which reduces the cost of analysis, the time for mobile phase preparation, increases the column and system's lifetime, all while closely matching physiological conditions. And just like the ion exchange chromatography example, we are once again using an infliximab innovator and biosimilar for SEC method scaling. So with the use of Premier columns, we've created a routine method for analysis of size variance using 1x PBS as the SEC eluent, which closely matches the physiological conditions at pH 7.4 and approximately 150 millimolar in ionic strength. So this 10-minute method was then scaled from the Acuity Premier system to the Arc Premier system. For SEC, although we are utilizing an isocratic gradient, parameters such as flow rate and injection volume still need to be scaled between systems to maintain the same column loading and column volumes for equivalent performance. So we evaluated the percent peak area for 5 replicate injections of infliximab to compare chromatographic performance between the original and scaled methods, and found that the peak area was within 0.05% for the aggregates and fragments, and shows that size variance can be resolved reproducibly across systems and column formats. Now, our last case study for method scaling is a peptide mapping evaluation of infliximab using a reverse phase gradient. So infliximab was reduced, alkylated, tryptic digested, acidified, and then injected onto both systems. So for complex peptide maps, they require a long, shallow gradient to adequately resolve the digested product. And this represents a challenging condition for a pump to reliably reproduce. In this slide, we chose 11 representative peaks to analyze the scaled method's ability to reproduce retention times, similar to the original method. So with using peak 6 as the reference peak, the relative retention time was calculated for the selected 11 peaks. Looking at the table, both instruments were able to reliably reproduce the peptide profiles with retention time standard deviation below 0.01, while maintaining separation selectivity between methods with relative retention times below 0.02 for quadruplicate injections. So both Arc Premier and the Acuity Premier systems have the ability to support LC-MS separations as part of any future or current development and manufacturing needs. So in this instance, the percent peak area was evaluated on the Acuity QDA, which is a mass detector that is designed with a small footprint and robust capabilities that can be adopted in manufacturing environments. So the chromatograms show the MS data represented as base peak intensity chromatogram, or BPI, and the extracted ion chromatograms, or XICs. Now, through the XICs, we can quickly filter the MS results to only display the selected masses of interest, as shown in the bottom chromatogram. Looking at the table, the QDA data for relative percent peak area aligns closely between both systems, with the difference being within 2% area, and the reproducibility of the MS results was also maintained with percent RSDs below 5%. So we will now move on from method scaling and talk about method migration. So the aim of this case study is to demonstrate how on a newer system, the Acuity Premier system with a fixed loop design can successfully migrate a method from its predecessor system, the Acuity UPLC Classic. while still delivering additional benefits in the analysis of monoclonal antibodies. So as part of the life cycle management process, ICH guidance recommends identifying and evaluating improvement opportunities to improve drug safety in a feed-forward fashion to continually expand the body of knowledge in the manufacturing of drug products. So this process can be applied to analytical methods as well as instruments as they become outdated with faster, more robust, and more sensitive technology that directly impacts consumer safety. So for the method migration case study, we will be looking at a peptide map of NISMAB 8671 as a representative challenge to the system in terms of its ability to deliver accurate gradient composition and selectivity of a complex mixture of peptides. However, unlike method scaling, we will not be changing any method parameters such as gradient, flow rate, and injection volume. We did, however, still evaluate the dwell volume between the two systems to see if an isocratic hold was necessary to maintain retention times between both systems. So the Acuity Premier fixed loop system was purposefully engineered to closely match its predecessor to facilitate easier migration of legacy methods. And since the dwell volumes were close enough, 150 to 160 microliters, we did not alter the start of the gradient after injection. So these similarities between the two systems translates to minimal or even no changes needed when using the same column format to achieve the same separation. This can be seen in this slide for a peptide map of the NISMAB digest reference standard run on both systems using the Acuity UPLC CSH column. So looking at the top right of this slide, an orthogonal plot of retention time for the 22 selected peaks is shown, and it exhibited a linear response with a slope approximately to 1 and a y-intercept close to 0, indicating that selectivity and absolute retention times were maintained across systems. On the lower right bar plot, we did a differential analysis of peak area between systems, and this shows that relative peak area was within 0.5% of each other, indicating that conservation of peak areas across systems was maintained. So now that we've established comparability between the systems when using the same column, we'll be now evaluating the performance and additional benefits that MaxPeak HPS columns bring to peptide mapping assays. So on this slide, we performed the same peptide mapping analysis on the Acuity Premier system using a CSH column that has with and without MaxPeak high performance surfaces. So looking at 4 representative peaks indicated by the red arrows, we can see from the bar plots that the MaxPeak Premier column was able to reliably and reproducibly perform the peptide mapping assay, demonstrated by the low standard deviation and retention time and peak area for the 4 peaks that we chose. However, we did see additional benefits of MaxPeak Premier columns when we looked at 4 acidic peptides. So these 4 peptides contained 2 to 5 acidic amino acid residues, which were either glutamic or aspartic acid. And all of these showed increased peak tailing and recovery issues with the stainless steel CSH column. So with the Premier column, not only did we see higher recovery for the acidic peptides, as indicated by the increased area counts in the bar plot, but the column also provides us the ability to see critical impurities, such as the deamidated species associated with the penipeptide, which is a peptide known to play a role in therapeutic efficacy of mAb-based drug products. So in summary, we have demonstrated that biopharmaceutical workflows can easily be migrated downstream to facilitate efficient method development and scaled to manufacturing environments while maintaining reliable and consistent results. So we've showcased this through our MaxPeak Premier product line that offers you flexibility to meet the analytical needs across your organization in development or manufacturing environments. I'd also like to take a moment and thank the scientists and marketing colleagues listed who have contributed to this webinar. And lastly, I wanted to mention that all of the data shared today can be found in the application notes listed on the left side of the slide. So these application notes include detailed information on sample prep, chromatography conditions, and contain additional data not shown in this presentation. And I've also linked a few supplemental application notes on the right side that provide additional tips for migrating methods. So thank you all again for attending the presentation, and I'll hand it over back to Jim.
Great. Powell, thank you very much for the wonderful presentation, and thank you again to all of our attendees for your time and attention. I did want to bring to everyone's attention here the upcoming episode that we have on streamlined monoclonal subunit LCMS workflow analysis for MAMs of biosimilars. This seminar series will be on October 12th, and it will be led by Jared Young from Similis Bio. So I encourage all of you, if you haven't done so, to go in and put your registrations in for that exciting event on October 12th. Before we get into the Q&A session, I wanted to pause here to have a couple poll questions that we'd like to share with you, and we certainly encourage all of you to put in your choices, which would be very valuable information for us to understand. Our poll question is around whether or not you've experienced mass recovery issues when working with biotherapeutics. In here, it's a single choice. Please put in your choice as never, not often, and often. And also, you'll notice that another pop-up has come up on your side of Zoom. Here, we'd like to get your answer here on whether or not you'd like to be contacted for more information about Waters products and solutions, and whether or not you'd like to have a Waters representative contact you. Please enter your answers as either yes, please contact me, or no, not yet. Great, thank you very much for that. And now we'll pivot to the Q&A portion of the webinar. And I see here that we've had a very active list of people, putting some great questions into the chat. The first one here is a question for you, Powell. Here, one of our attendees has asked about what is a fixed loop system. Can you please help all of our attendees understand what the fixed loop system is?
Yeah, so in the presentation, when I was referring to the fixed loop system, I was talking about the injector design used in the auto sampler. So currently the Acuity Premier System auto sampler has two configurations: the fixed loop, which we used for our peptide mapping data, and the flow-through needle design, which we used for the IEX and SEC data sets. So the fixed loop design is the preferred design if you require the lowest dispersion for any high resolution separations. And the needle is also offline, so it tends to have the fastest cycle times since the needle can be cleaned and prepared for the next injection during analysis. There are also 3 injection modes to choose from for optimal performance. However, on the other hand, the flow-through needle design offers the widest injection volume without the need for a configuration change and can offer you more flexibility and ease of use.
Yeah, thank you very much, Paul, for that answer. Here, there's another question that I could take. The question is, can the Premier system handle additives and modifiers that are typical for bioseparations? Here, the answer is yes. Before these systems were launched, robust testing was performed to understand, the ranges of modifiers, eluents, and mobile phases that could be handled by MaxPeak Premier systems. So here, all of the typical eluents, modifiers, and mobile phases that you would use in bioseparations, whether it's 100% methanol, HFIP, sodium acetate, or phosphate buffers, those concentrations that are common for those applications, the Acuity Premier and Arc Premier system can handle those conditions. And if you need additional information, we do have a lot of resources available on waters.com. One key resource here is the Solvent Considerations Guide, which lists out all of the recommended limits for particular mobile phases, eluents, and modifiers. So, yeah, thank you very much for that question. Paul, here's a question for you. When would you recommend a quaternary or binary pump for monoclonal analysis?
Yeah, so it really depends on the application that you are running. So the largest difference between the pumps is that a quaternary pump can blend solvents, whereas a binary pump cannot. So if you're doing a lot of method development, the quaternary pumps give you more flexibility in evaluating characteristics that can improve separation of your analytes. So for example, the IEX data that we went over today, we utilize AutoBlend Plus with the quaternary pump to formulate several different mobile phase compositions from concentrated stock solutions, so we could quickly scout how pH and ionic strength affect our separation. On the other hand, binary pumps are known to provide the best performance for long, shallow gradients and are ideal for peptide mapping analysis, where methods are often over an hour long. Another thing to consider is if you are running the same method over and over again, a binary pump has a lower dwell volume, which decreases your analysis time for higher throughput capabilities. So really, in summary, there is a place for both pumps in MAB analysis.
Great. Thank you, Paul. And thank you again to the audience for that wonderful question. And the last one here we have in our question and answer chat box, I can take this one. This is around the difference between an Acuity Premier and an Arc Premier system. Great question. So at the highest level, customers choose an Arc Premier or an Acuity Premier based on operating pressures. So the Arc Premier system has a maximum operating pressure of 9,500 psi, and the Acuity Premier has a maximum operating pressure of 15,000 psi. And along with that, the column chemistries that pair with these systems, they vary. So for the Acuity Premier system, that is always paired with sub 2 micron Max Peak Premier column chemistries, and the Arc Premier system pairs with 2.5 to 3.5 micron column chemistries. And then the last key difference between the two is around the mass spectrometers that are configurable with the systems. So the Acuity Premier is configurable and supported with all of Waters mass spectrometers. The Arc Premier system pairs with single quadrupole mass spec systems along with the lower end mass spec systems that we offer in our portfolio. So those are the three key differences between the two different platforms. Again, thank you very much again for that question. And I do see here that all of our questions have been answered. So, if there are any additional questions that you may have, please feel free to reach out to Powell or myself at any time with any questions you may have. And I'd like to take this time to thank Powell for the wonderful presentation. Thank you very much, Powell. And I'd like to thank all the attendees for joining today's episode 3 of the Biosimilar webinar series. And again, as a reminder, there will be a 4th episode coming up on October 12th. So I encourage all of you that are in attendance today to register for that event next week. So with that, thank you again for your time and attention. And Powell, thank you again for the great presentation.